Welcome back to AP Biology, helping you study the study of life.
Hi, and welcome back to our channel where we help you study the study of life. Today, we're going to talk about DNA replication. So, what is it and why is it important? DNA replication is the copying of DNA, and this is important for mainly two processes, mitosis and meiosis. Mitosis is the repairing of cells. Like when you get a cut, mitosis is the new growth of tissue. In mitosis, one cell will form two identical daughter cells, and if the DNA did not replicate, every new cell would have half the amount of DNA, or half the number of chromosomes. And when those half number of chromosomes were to replicate themselves, the number would slowly just be cut in half to eventually where there would be little to none DNA information within these cells. And so you can see how it is so important that the DNA replicates so each cell has the correct number of chromosomes. Next, meiosis is pretty similar, but it is a splitting of cells during reproduction. And during reproduction, the offspring are receiving DNA from the mom and the dad, which is why there are essentially two splits. One that makes two cells and one that makes 4 haploid cells, meaning it has half the number of chromosomes. And if the chromosomes did not duplicate at the very beginning, each offspring would be short chromosomes and they would be short genetic information. So one thing that we quickly have to mention is the structure of a nucleotide, and it is made of a sugar group, nitrogenous base, and a phosphate group. And scientists have numbered the parts of the nucleotides, but the numbers we are concerned with are the 3 and the 5, and we refer to them as the 3' and the 5'. And as you can see in this picture, the 3' end is on the sugar part and the 5' end is on the phosphate group. Okay, so let's real quickly review that certain nitrogenous bases pair with other nitrogenous bases. Guanine always pairs with cytosine, while adenine pairs with thymine. But remember, in RNA, there is no thymine, so adenine will pair with uracil. And the reason that these bases pair with each other is because of their sizes. Adenine and guanine are both purines, which means they have 2 rings. And cytosine, adenine, and uracil are pyrimidines. They have 1 ring. A purine always pairs with a pyrimidine. This way, the length between the 2 DNA strands is always the length of 3 rings. And one way you can remember which one pairs with the other is the phrase cut small pyramids. The letters in cut stand for cytosine, uracil, and thymine, and these are the pyrimidines. So you can kind of see the word pyramid and pyrimidines, and then you can remember that the other ones are purines. Also note in the next few pictures, there are going to be 2 red molecules. One is a phosphate group, part of a nucleotide, and one is the base thymine. The red molecule on the outside of the DNA strand will be the phosphate group, and the one in the middle is the base thymine. Okay, so here you can see we have a nice DNA strand. It has 2 strands, a leading strand and a lagging strand, and we'll talk about that more later. The red and white are the sugar and phosphate groups of a nucleotide, and the red, blue, green, and orange molecules are the nitrogenous bases of adenine, guanine, cytosine, and thymine. And you can see the 5' end and the 3' end. Also notice how the strands are backwards in a way. how on the lagging strand, the 3' end is on the left, but on the leading strand, it is on the right. And the 5' end on the lagging strand is on the right, while on the left, it is on the leading strand. And this will be super important later. Okay, so there are lots of enzymes that are involved in this process of replicating DNA, but the first two are Topoisomerase and helicase. And we know they're enzymes because they end in ase. So Topoisomerase unwinds the DNA strand, while helicase unzips the DNA. So if you think about DNA as being a zipper, DNA helicase is just unzipping the zipper. So DNA helicase will only unzip part of the DNA at a time, and you can see here the replication fork. So the next enzyme that comes along is RNA Primase. And the reason we have to start with RNA is that the DNA needs something to latch onto. We cannot just immediately start copying DNA into DNA. So RNA Primase will read the DNA strand from the 3' end to the 5' end. So on the lagging strand, it will start on the left and go to the right. And on the leading strand, it will start on the right and go to the left. Since we are copying each individual strand with its complementary strand, the RNA will build from the 5' end to the 3' end. And in these pictures, the RNA is the pink molecules. So now that we have a place for DNA to attach to, thanks to RNA primase, another enzyme will begin working, and the next enzyme is DNA Polymerase III. DNA Polymerase III will attach to the RNA primase on both the leading and lagging strand. On the lagging strand, DNA Polymerase III will read from the 3' end to the 5' end, which is from the left to the right. But remember that it is building the complementary strand. So it is reading from 3 to 5, but it's going to build from 5 to 3. So the strands are anti-parallel. And the leading strand will attach to the RNA also and read from the 3' end to the 5' end, which is going from the right to the left. But it also is going to build the complementary strand, so it's anti-parallel. So it'll build from the 5' end to the 3' end. So again, read from 3 to 5, but build from 5 to 3. So now that we have copied the part of the DNA that's unzipped, we can then move on to the next step, which is the unzipping of more DNA. And this is where helicase will come in and unzip more. So after more is unzipped, we can now see that on the leading strand, there is DNA that DNA Polymerase III can attach to. So RNA primase is not needed again on the leading strand. So DNA Polymerase III will keep reading 3' end to 5' end, right to left, and build the complementary anti-parallel strand by building 5' end to 3' end. But on the lagging strand, there is nowhere for DNA Polymerase III to attach to. So RNA primase adds RNA on the lagging strand. So now that I have a place for DNA Polymerase III to attach to on the lagging strand, thanks to RNA primase, it can go in and read the strand from 3' end to 5' end, which is left to right, building the complementary anti-parallel strand 5' end to 3' end. And once the DNA Polymerase III has done its job on both the leading and lagging strands, we will then repeat the process with DNA helicase unzipping the rest of the DNA strand. Then RNA primase will add to the lagging strand again, while DNA polymerase III continues copying DNA on the leading strand. Okay, but now if we look at our strand, we have a whole bunch of RNA, but we need it all to be DNA. So this is where Polymerase I will come in, and he will go in and replace the RNA with the DNA. So as you can see here, the RNA is being replaced with DNA on the leading and the lagging strand. And after we replace all the RNA, we notice that on our new strands, there are places where they're not connected and there's broken places. And this is because when we replace the RNA, DNA polymerase I didn't connect the strands. And so we need another enzyme for this job, and this is ligase. Ligase is the enzyme that will come in and glue all of the strands together. And these strands, these broken pieces, are called Okazaki fragments. So after ligase does its job, the very last enzyme that is needed for this process is telomerase. And telomerase adds the nucleotides TTAGG to each 3' end of the new strand. And this is just a way to protect the new strand from chromosome erosion, which happens with old age. Okay, so to review, DNA replication is the copying of DNA. This is important in the processes of mitosis and meiosis. To start the process, helicase and topoisomerase will unwind and unzip the DNA strand. Next, RNA primase lays down RNA. DNA polymerase III will then come down and lay DNA. Next, polymerase I will replace the RNA with the DNA. Ligase will then connect all of the Okazaki fragments. And finally, telomerase will lay down TTAGG for protection from chromosome erosion. Well, that's all we have for today, folks. I hope you found this video helpful, and thank you for tuning in. Go to our channel for more videos that will help you study the study of life.